This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 534, recorded on February 8th, 2019. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello there, Vincent.
Vincent?
No, I meant Vincent. Hello there, Vincent.
You want me to edit it?
Would you?
Welcome back from France.
Thank you. Yes.
And the weather has cleared up.
It's très bien.
It was cloudy and raining when I came. Now it's sunny, but it's windy, right?
Windy, but nice temperatures in the 50s. It's incredible.
Also joining us from southeastern Michigan, Kathy Spindler.
There you go.
Hi, everybody. We had the sun kind of, it's bright, but there's clouds, but there's some little tiny patches of blue, which is really nice, but it's cold. I think you're going to get cold weather soon, Vincent.
Oh,
Think so? That's fine.
Another Polar Express?
No worries. It's,
it's 18 Fahrenheit right now,
Eek.
which is minus 8 Celsius.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and it's temporarily gorgeous here, and also the forecast says it's going to get chilly.
Right.
And from Madison, New Jersey, Brianne Barker.
Well, in the long term, you might have cold weather, but here it's blue skies and beautiful. So short term, It'll be good.
That's right.
Hi, everybody.
Hey, Brianne.
So people think we don't know how to interact if we don't have the weather.
We
but
don't.
We
Right.
do. We can. It's just that the weather.
But we won't.
Yeah, we interact for 5 minutes before the show starts.
Yeah, we do.
But we don't even talk about the
Sometimes
weather.
But if we had no weather, it would leave a vacuum at the beginning of the show.
No. Sometimes we talk for 10, 15 longer minutes. Not all the time.
This is true.
And then Alan says, okay, can we do a podcast?
Yeah.
Wants to get us going.
Maybe you should call our group Cloudy with a Chance of Meatballs.
Oh, you know how long you've said that on different podcasts.
I guess.
You know,
Have I really said that?
you never come up with new jokes.
Oh, I'm an old guy. What do you want? Let me just get my specs. I'll be right back.
What, your glasses?
Yeah, I have to get some reading
There's
glasses.
one right there. Right there.
Right?
Well, you can't see, I guess. See the Parasitic Diseases book?
Oh, yeah. Okay, I got it.
And then when you come back, I'd like to know how France was, just briefly. Good or bad?
It's très bon.
Was the food great?
Fantastic.
Okay.
Yeah, it was great.
All right. We have a couple of meetings to tell you about. European Congress of Virology for 2019. The abstracts are due March 1st. That's ecv2019.com. The ASM Clinical Virology Symposium, May 5th through 8th, Savannah, Georgia. Abstracts due March 8th. asm.org/CVS. The Intel International Science and Engineering Fair. That's in Phoenix, May 14th through 15th. They need judges in 22 scientific and engineering disciplines. Dickson, would you like to be a judge?
I've done some of that work already. Thank you. I've had enough.
All right. Anyway, we're going to put a link in the show notes to that. And that's it. Dickson, you have any announcements?
No, but I'm still having a good time. So what can I say?
All right. So let's jump right in here. We have two papers, a snippet and a paper, and they're not related, except that they deal with viruses. And one's an RNA virus, one's a DNA virus. And the snippet is all about virus structure. We just did our virus structure lecture Monday. So I'm primed for this. And I just rewrote the coronavirus chapter for Fields Virology along with Amy Rosenfeld.
Yeah.
And we did a lot of the
Structure.
topic that's the point of this paper. This is a letter in Nature. Calcivirus VP2 forms a portal-like assembly following receptor engagement. Very exciting.
Ooh.
Conley, McElwee, Asmi, Gabrielson, Byron, Goodfellow, and David Bella. I have interviewed both Goodfellow. He's a good fellow.
Bello is a good fellow.
And Bella, you can find them online on YouTube. And this comes from the MRC University of Glasgow Centre for Virus Research and University of Cambridge. And so this is a structural group there, Dave Bella's group. And caliciviruses, as everyone knows, cause gastroenteritis.
Let's not exclude people who don't know.
They do know that they cause gastroenteritis.
I thought we were going to say they were an important part of Game of Thrones.
No, I don't. What's that about?
One of the characters has a queen, and her title is Khaleesi.
I've never seen Game of Thrones, so I wouldn't know. Never watched it. But everyone knows that you can get sick, maybe not from this virus. You know, it has been called the cruise ship virus, but
a norovirus is the
Calicivirus family includes noroviruses.
Okay,
it all right. Noroviruses. All right. And that because of that,
these are unenveloped RNA viruses.
They're in my view, they're imitators of picornaviruses.
Wouldn't want to be picornas?
They're about the same size, same kind of shell. Genome is 7.5 kb. And here's the thing: these viruses, at least the noros in humans, the disease is called winter vomiting disease. They have not figured out how to replicate in the gut without causing disease, whereas poliovirus, for example, replicates in your gut with zero gut pathology, no gastroenteritis, is efficiently transferred to persons after. It is just the best virus in terms of gut replication.
Well, that said, norovirus has a pretty good strategy that relies on causing its pathology.
Yeah, that's another way to do it, which just goes
causes
to show
this projectile vomiting that then spreads the virus.
It just goes to show there are more than one way to skin a cat. And speaking of cats, there are feline caliciviruses, and that's the topic of this paper. It's a good model system because they grow well, and the receptor's been identified. So these are viruses with plus sense RNA genomes, about 7.5 kilobases, and they are packaged in a shell that's built with icosahedral symmetry. Dickson, right there on the table in front of you, there is a shell built with icosahedral symmetry.
I would never doubt you for the least.
Yes,
so, because, as Alan mentioned, these are non-enveloped, this brings up to me what's the whole point of this paper. And this is that for the non-enveloped viruses, they have to have some strategy by which they get their nucleic acid into the cell beyond a membrane. Right. And it seems like each one that we find out about has a different way of doing it. Adenoviruses do it differently from the papillomaviruses. We covered papillomavirus in a TWiV in December, where they stick a protein out through the endosome, and then that gets pulled by the retromer. And so the way that we're going to hear about today is just a fascinating new way for an RNA virus to do this.
Right. And for enveloped viruses, just to complete the circle there, that their solution is very easy, that once they fuse their membrane with the cell membrane or with the endosomal membrane, then the viral capsid is in the cytoplasm. But an unenveloped virus gets taken into a lysosome or an endosome and is then stuck there inside this membrane that it has to penetrate, and that's what we're going to talk about.
Yeah, I always say it's conceptually easy for these membrane-containing viruses, right? You fuse.
Yes.
But for the protein shells, though, so what happens, and as Kathy said, there are lots of ways to do this, and this is a new way, which may be perhaps more general than we know at the moment.
And it's really cool.
Very cool.
It is.
And so these capsids are made most of a major capsid protein, VP1, 180 copies, by the way. This is a T equals 3 shell. The main capsid protein, VP1, and then there's a minor protein, VP2, which is not as far fewer copies than VP1. Far fewer. And that's going to be important.
But it is not minor in importance.
Not minor in importance, because, in fact, if you mess with it, you lose infectivity. So it's really, really important. Now, the receptor for feline calicivirus is tight junction cell adhesion molecule called junctional adhesion molecule, JAM-A. And the junctional adhesion molecule is also a reovirus receptor. Feline JAM-A. Typical transmembrane short cytoplasmic tail, transmembrane, and 2 immunoglobulin-like domains. And Dickson, how many Ig-like domains does the polio receptor have? 3.
Don't do this.
3.
To me. Please don't
3.
do this to me.
How many times of kinds of genomes?
7.
Yeah. So this JAM-A binds to the capsid, the outer capsid. Capsid has been shown to interact precisely before. People have visualized this. It causes conformational changes in VP1 assemblies on the capsid, and these are probably precursors to genome release. And that the way this happens is the virus binds cell surface receptor. It's taken up by endocytosis, and then low pH in the endosome is required for the RNA to get out of the particle. Yeah.
What part of the virus actually forms the pair for the ligand and the receptor? What is this VP1?
VP1.
1.
But as you will see,
okay,
there's some role for VP2, a very important role. Yes.
My question would have been, what happens if you flood the host with VP2? Do you prevent infection?
I don't know the answer to that.
Okay.
And so they didn't do that here.
All right.
But I would bet it doesn't do anything.
Nothing.
All right. So they wanted to look at the structure of the virus bound to JAM-A. And the term that they use is decorated, and that's the structural biologists. They all call it decorated. Virus is decorated with receptors. So it's like a Christmas tree, I guess. They do cryo-electron microscopy. And, of course, when you do that, you take lots of pictures, and you look at many, many pictures and do icosahedral averaging to create an image, a three-dimensional image of the particle. And they noticed that when they added the receptor to the calicivirus, where the receptor and VP1 touch on the capsid, the density was blurred. In other words, icosahedral symmetry is broken by the conformational changes that occur when the receptor engages the virus particle. So then they backed off and said, let's do some non-icosahedral methods for figuring out what's going on here. And this is very interesting. In many viruses now, people are backing away from icosahedral averaging, and you're getting information that you didn't see before, where you assume that everything is icosahedrally symmetric. Well, it's not the case. And as you'll see in this paper, you reveal some very interesting things.
Yeah, they don't fit into the shapes that we were making them fit into.
No. In fact, the whole particles now are done with this. And the one I just saw that's very interesting is a paper was just published on a flavivirus where the capsid inside the envelope could never be visualized well at high resolution. So they turned off icosahedral symmetry, and then they saw it, and they could see that it's beautiful. Anyway, back to this calici. When they do this analysis, they see a brand new structure protruding from the capsid comprised of 12 fingers of density around the threefold axis. And within that density, there's a portal that goes through the capsid.
Sounds like a pore form.
Yeah. And this is at only one place in the capsid.
At only one place. Right. And that's the cool part of this. It's only at one place. Yeah. And you didn't see it before. And so portals are known to be on herpes simplex viruses. And here I'm going to show Dickson this keychain I have.
Yes.
Inside of it is, so there's the membrane. I can open the membrane. And inside is a herpes capsid. At one end, on one fivefold axis of symmetry, there's a portal, a preformed portal. And that's what docks and releases the genome. But for this calici, you don't see it ahead of time. And then when it hits the receptor, it forms at one threefold axis in the particle.
The receptor pulls its fingers.
Receptor pulls its fingers. So it's a single portal at a unique threefold axis, and it's composed of VP2, 12 fingers of VP2, and that's all the VP2 there is in the particle.
Wow.
And the pictures are beautiful. They show the particle at the threefold axis, and there are 12, of course they've colored them nicely, fingers of VP2. So the receptor binding has caused conformational changes, and these VP2s have swung up. So normally they're buried. They have swung up, and their end termini are hydrophobic, which will allow them, of course, to insert into the membrane. And because there's a portal there, presumably the RNA can pass through.
Wow.
And that's really This pore is a nanometer in diameter.
So I had to look up how that relates to the size of a nucleic acid.
Yep.
And looks like a double-stranded DNA is about 2 nanometers.
Mm-hmm. What's the same?
So one nanometer. And I would guess that it's probably not strictly 1 nanometer. I want to talk a little about the Phi-X findings, and we can relate to that. But
yeah, so it's tight, right?
Mm-hmm.
It's a tight fit for that RNA, but it probably could get through it. Of course, there's a static structure. You don't know, might change a little bit. All right, so basically you have 12 copies of VP2, which are VP1 is engaged by the receptor. It causes conformational changes in VP1, which then allow VP2 presumably to extend the normally buried N-termini, which is hydrophobic, and you make these long alpha helices. And what's really amazing is that this is so similar to the low pH changes in the hemagglutinin of influenza virus. As it passes through the endosome, the fusion peptides are at the N-termini, which are buried near the viral membrane. At low pH, they swing up, just like these VP2s are swinging up. It's a common theme. It's really cool.
Well, they're addressing a common problem.
They are.
The viruses need to respond to a low pH and open themselves for uncoating.
Right. All right. So that's the model, right? The receptor binds, JAM-A binds to VP1. It causes conformational changes. They think that the receptor dimers are disrupted. The virion is enwrapped within the endosome. You get more JAM-A molecules binding that trigger conformational changes. You get this formation of a portal, and the N-termini of VP2 come out and form a channel, and the RNA genome comes out through the channel. And so they say these features have not previously been described in small RNA-containing viruses. So I wrote last night to Dave Bella, and I said, wait a minute. Okay, there is a portal in herpes. That's fine, but it's a DNA virus. But in picornaviruses, the last few years, people have been seeing portals in the particles for polioviruses and Coxsackie viruses and others. And so I said, what do you think about that? And he said, well, this is a portal made of this specific protein, VP2. For the other viruses, it looks like the viral proteins rearrange, and they form. So this is unique in that you have a dedicated protein, VP2, to forming this portal. And then the question arises, well, for these viruses, all right, so you make a portal, beautiful, makes a pore in the endosome membrane. How does the RNA get out? Because where is the end that has to come out first, right? It could be anywhere in the particle. So it has to be near where the portal is going to form. There's no other way it would work. So on assembly, the portal has to be
right, because this portal is not wide enough to admit a folded piece of RNA.
No, it's got to go through.
It's got to be the single. Through. It's got to thread through. So
the portal has to assemble near where the end of the RNA is. So the exit point has to. So this is what Dave said. The exit point for the RNA has to be established at assembly, not at receptor engagement. So let's say wherever the virus hit a receptor, that's where the portal formed. Well, what if the end of the RNA is on the other side of the virus?
Then you'd have a breach birth. Yeah,
exactly. So, and I said, well, for the picornas, there's no evidence, but there must be preformed portal forming areas that we haven't seen. And he said that's because we only look by icosahedral averaging of these particles. And if you back off, you might see them. Now, Jim Hogel's latest paper, he looks at the conformationally altered particle. Alan will remember the 135SA particle.
Oh, yes.
You can find a portal in that. But what about if you go to the native particle and don't do icosahedral averaging? Would you see some one threefold axis that's slightly different? So it's interesting.
Now, on the size of this portal, do we know that that's a fixed size? I mean, crystallographers always tend to think in terms of everything being rigid, but these are flexible proteins. Could that actually open up wider?
I don't see why not.
Yeah, I suspect that's probably the case.
Because if that were the case, then it wouldn't be so critical to get the tail of the RNA in one place.
It still seems like it would have to be close. And Vincent, don't we know that the 3' end of the genome exits first?
Yeah, for rhinoviruses it does, so I assume it's the same for polio, right?
Right, there is an order. Okay.
So somehow it has to get near the place that's going to be a portal, right?
Yeah, so, maybe maybe for these caliciviruses, there's something that associates with whichever end is going to come out. And, maybe it's a 3' end, and that's where because that was my question is how does it know where the portal's going to form?
Right.
Well, for herpes, you have one preformed portal, but then the DNA goes into the capsid through the portal. So that's easy, right? Because then the end ends up right there, but we don't know. And I thought, well, maybe it's VPG, the protein linked to the 5' end, but it's the wrong end. It's the 5' end.
So I have a question. When I wake up every morning and I do my duty in the bathroom.
TMI.
No, not enough information. And I go to brush my teeth. I take out the toothpaste. I remove the cap.
Yeah.
Why doesn't the toothpaste come out, Vincent?
It's not under pressure.
Wait a minute.
These RNAs are under pressure.
Well, you didn't say that.
Huge pressure.
You didn't say that. Okay.
Yeah, a lot of pressure.
Okay. And what is the basis for the pressure?
Well, the fact that they've got the entire viral life cycle riding on them, that's a lot of pressure.
Okay, I give up.
Well, so for some viruses, for some DNA viruses in particular, the portal of herpes viruses and bacteriophages, there's a pump that winds it in under pressure. It requires energy. Those have been well studied. But for these RNA viruses, we don't know.
Okay.
But they are under pressure. It's been measured by atomic force microscopy.
Okay.
And you can see the pressure that's, and so it comes right out when the capsid is breached.
Got it.
So Kathy, you mentioned Phi-X?
Yeah, I wanted to talk about Phi-X because they call it out also.
Yeah.
And tailed phages, we have some understanding of how the DNA or the, I don't know if there's any tailed RNA phages. Anyway, how the nucleic acid comes out. But for Phi-X, which has a single-stranded circular DNA, there's no tail. It's an icosahedron, non-enveloped. Mm-hmm. And so this paper that the last two authors are Michael Rossman and Bentley Fain. And they found that the H protein, which is known as the pilot protein, oligomerizes to form a tube. And so there's 10 molecules of this, somewhere on the order of 10 to 12 in the virion, but 10 of them oligomerize to form this tube that the DNA can go through. And the diameter of this H tube is 22 nanometers. Now, the single-stranded DNA, being a circle, has to go through one strand basically in each direction. So it has to be at least 2 strands wide. But 22 nanometers is plenty. So that was a really cool Nature paper from 2013. And because my first work on Phi-X174 was with the H protein, I was really fascinated by that. So that was.
So Bella says, he wrote to me, I think inherent asymmetry is likely a feature of most or all icosahedral viruses. And he also sent me a link to, so he said they had to shorten this paper a lot to be a Nature letter. And he said there's an unabridged version over at BioArchive. I'll put a link
yes,
in the show notes where he does more extensive discussion, because I said, you mentioned in one sentence this business about portals. He said, yeah, we had a lot more on that in the original. So
okay, that would also be good because this is not an open access paper.
Yeah.
Which is too bad because these are gorgeous pictures.
Yes. Are those the pictures are in the preprint as well?
Yeah, and movies as well, right?
Great. The movies are amazing. Yeah, cool. There are there are 9 of them, and I highly recommend. And some of them are only a minute, but some are like 5 minutes long, and they're just amazing. I recommend 2, 3, and 5. And in 5, they're showing the contact interface between the viral VP1 and the JAMA receptor, and they have each of them colored, and then they tell you that they're going to show you the contacts. And so you're looking at the molecule and you're thinking they're going to pull those 2 things apart. And they do, and they rotate them towards you so you can see the contacts, and they're all color-coded. It's just fabulous. They must have spent hours making these movies.
Mm-hmm. So art and science do come together.
Yeah.
We had an email about that.
Yeah, I know. I got an email from Kathy. Thank you, Kathy, again.
Because we know you wouldn't listen.
Oh no, I don't listen. I look.
All right. Any further discussion? Very interesting stuff.
Yeah.
It's an aspect of virology that we used to do way more than we do now.
Yeah. Does this suggest interference possibilities? Now that you know the mechanism?
Yeah, but we can do that other ways.
No new information with regards to strategies for interfering with attachment.
No, you don't need to. You don't need to.
Okay.
Well,
just
not
asking generic.
Not immediate. I mean, the application for this is not immediately obvious.
Okay.
But the more you know about the virus, the better off you are in designing things.
No question.
Right. Because you could imagine if you if you were looking for an antibody to form against this VP2, it probably wouldn't happen because it's mostly hidden until the moment when it's already bound to the receptor, at which point an antibody would be too late.
I'd make it an IgA molecule stuck in the mucous membranes in the gut tract.
You can argue. You just need to know things, and stuff will come out of it, Dickson. But immediate benefit does not have to be.
I was just asking for the collective knowledge of the virologist in the audience.
Understand. I actually didn't look at this at all because there's some fundamental principles here that are really important about disassembly, right?
And basic biology of virus.
But it will lead to something.
Got it.
For sure.
Yep.
And if nothing else, it's also further evidence that people need to think about whether they're using the icosahedral fitting when they look at their structures.
Mm-hmm. Yeah, I mean, that was the original breakthrough. You do icosahedral averaging, you get these beautiful structures, and now we have to back off. You have to use it, shall we say, judiciously.
There you go.
Judicious use of icosahedral averaging. Boy, if that isn't a geeky, nerdy thing.
Yes.
It sounds like a review article title.
It would be. All right. For our main paper, it was suggested by listener Michal. Dear TWiV crew, first, I'd like to thank all of you extremely likable hosts for a great podcast and fascinating discussion. One of the interesting topics in 2018, in my mind, was the discussion in TWiV 498 about methyl 6A modifications on viral RNAs, and I thought you might be interested in this cool work from the lab I work in, published in Nature Immunology, which shows that M6A modification controls the innate immune response to viral infections. So he sent along a paper that we'll discuss, Nature Immunology, but he also reports similar findings were reported also in this paper in Genes in Development from Ian Moore's lab. Happy New Year, Michelle, staff scientist at the Department of Molecular Genetics, Weizmann Institute of Science. And yes, this is pretty cool. I decided to do the Nature Immunology paper because Michelle sent it to us. The other has similar findings. There's some differences which we can point out. But this one is M6A modification controls the innate immune response to infection by targeting type 1 interferons. I have to say, Brianne, I delayed this to this week so you could participate because you weren't there last week.
Good. Thank you.
And I thought you would like it.
I did like it. And I spoke to some high school students earlier today and was telling them some important features of immunology. And I feel like this paper actually talks about some of the same features I was telling them about.
Great.
So this is, let's see, from the Weizmann Institute in Israel. The first author is Ronnie Winkler, and the last author is Noam Stern Ginosar.
So.
And I've read you the title. And this has to do with methylation of adenosine at the N6 position.
And it's not just the Weizmann. There's also Chaim Sheba Medical Center in Ramat Gan and Tel Aviv University and Universitat Duisburg-Eisen in Essen, Germany. Sorry, wanted to get the institutions in there.
Yeah, so the N6 position is on the base. There can be many modifications of RNA in different places, but this is a very well-studied one. And some of you may remember episode 498, Stacy Horner was on that. That was recorded in Atlanta. And she talked about M6A modification. And there are a couple of words we have to tell you about. And I wrote these out because I knew Dickson would ask. So they're enzymes that put on the methyl or read it to see if it's there. So there are writers and readers and erasers. So the writers are the enzymes that methyltransferases that put the methyl on the RNA, like METTL3, METTL3, methyltransferase-like protein number 3. That's what that stands for. Dickson, it's the catalytic subunit. There's a cofactor called METTL14, and there's another cofactor called WTAP, which stands for Wilms tumor 1 associating protein. I knew you would ask that.
I would.
So METTL3 is a writer of M6A modifications. Then we have readers like YTHDF1, YTH domain family member 1. I could not find out what YTH stands for. Did anyone have any success?
I didn't try.
I could not.
No.
I tried, and I said, Kathy will figure it out. YTHDF1, 2, and 3. Okay, so those are readers. They say, oh, there's a methyl here. Let's do something. Or there isn't a methyl and we're not going to do anything. And then you have erasers, demethylases, like ALKBH5. And that stands for alkylated DNA repair protein homolog 5, or FTO. That's another eraser, fat mass and obesity associated protein. That's one of the first mRNA demethylases that were identified. Okay, Dickson?
Yep.
That was all for you.
Thank you.
And so these, obviously this area, this methylation of this position and its consequences have been studied extensively. And this modification, what does it do? It regulates a whole bunch of things like decay of mRNA, which will come up in this paper.
It is the most abundant internal mRNA modification, they say. So this is something that the cell uses for all kinds of stuff. You need to keep track of this RNA or make sure it gets degraded or make sure it doesn't get degraded. You modify it or unmodify it with this M6A modification.
And of course, viral mRNAs are also modified.
Right.
And as Stacy Horner told us, it can have diverse effects. It can increase replication. It can decrease replication. There's no consensus. Right. But what they look here.
It all depends on context.
It all depends on context. And here, as you will see, it turns out that this modification can regulate the production of interferon-stimulated genes. All right. So the bottom line is that methylation is a negative regulator of interferon production because it dictates fast turnover of interferon mRNAs. All right. So the virus used here is human cytomegalovirus, DNA-containing virus that replicates in the nucleus. And it turns out that when you infect cells with this virus, the M6A writers and readers are induced. The writers and readers are induced. And
right, so the virus is doing something with M6A.
And there, that statement is as much as we're going to know about that. Because I think in the end, we'd like to know how they're induced, but we don't know that.
Right. But the other thing that's really important is at this point, we know they're induced, but we don't know what they're writing and reading on.
No.
Right.
Are they writing on a transcript that's important for the virus, a host transcript? What?
Right.
It could be not known.
So now you can ask, all right, if we take away the writers and the readers and the erasers from the cell, what happens to virus infection, right? And they do that in this paper with CRISPR-Cas9 disruption of the genes. And in the other paper, they do it with small interfering RNAs. But the same idea. You want to know what's going on. So they take out the genes encoding these proteins, then they infect with human cytomegalovirus. So when you deplete writers or readers, you get a reduction in virus titers. Okay? Writers and readers, you get a reduction in virus titers. If you take away the erasers, you get an elevation in virus titers.
So we just had a mnemonic. Writers, readers, reduction. We'll start with the R sound.
Yeah, the 3 R's.
That could be. All right? So somehow this writing and reading and erasing of methyls is impacting the yield of cytomegalovirus.
And specifically, cytomegalovirus does better when there's more M6A, with more writing and reading of
right.
M6A.
This could be confusing, Dickson. can be because it confused me. Because when you have all these negatives or positives, so more writing of and reading of methyl, the virus goes down. If you erase the methyl, the virus goes up. Okay. All right. So what's going on here? You can
wait. No, more writing and reading of methyl, the virus stays up. When you take away the writing,
yes.
take away the writing and reading of methyl, then the viral titer goes down.
I wasn't confused, but you were.
I was, yeah.
Yes.
Take away the writers and readers, you go up. Down, you go down. You take away the eraser.
I didn't know enough to be confused.
The virus wants the RNA methylated.
No, virus doesn't want anything.
I know the virus doesn't want anything, but it benefits the virus to have the RNA methylated and read as methylated.
That's better. That's much better.
And the other thing to note is that sort of from here on in the paper, they really focus on METTL3, and that's a difference with the other paper.
And that, of course, brings up the pedal to the metal.
Yes.
Dickson's, because you didn't come up with it, you're making a face.
Probably.
So here's a little side technology here. You could look at genome-wide M6A methylation of RNAs. Okay, this is great. You can take total RNA from a cell. Okay, you can break it up into small bits. You can immunoprecipitate it with an antibody to the M6A modification. So then you're immunoprecipitating RNAs that have M6A on it, and they're about 100 to 200 bases long. And then you can sequence them, high throughput sequencing. So you can do genome-wide analysis of what mRNAs have or do not have M6A. And in fact, I was reading, you can do it to single nucleotide resolution by a combination of experimental tricks in bioinformatics where you can predict where the methylation site is. So you could say, look at all the cell RNAs under certain condition or viral RNAs to see how the methylation pattern changes, what genes tend to be methylated, and so forth. That's very cool. So that's genome-wide M6A profiling. So they say, okay, what's going on in the cell? They see, okay, there are 25 viral RNAs that have enriched methylation specific to wild-type cells, but not METTL3. Okay, METTL3 being the writer, right? So that makes sense. But it doesn't seem to make any difference. There's no changes in the expressions of these methylated. But when they look at cell methylation, they found a big induction of interferon-stimulated genes when you deplete METTL3, when you deplete the writer. Actually, it's better if we use writers and readers and erasers, right?
Yes.
Yes.
Deplete the writer, so we could deplete Alan Dove.
Right.
And you get more ISGs. So many jokes here, right?
Yes.
So somehow this methylation, remember, which is turned up by HCMV infection, is regulating the interferon response because ISGs are turned on by interferon. And so they look at that directly. They have an inhibitor of the downstream signaling events downstream of the interferon receptor. It is a drug called ruxolitinib. It's an
AB stands for, at least.
No, it's not an AB. It's a nib. It's a drug.
Nib. Oh, you said nip. I thought you said ib.
It's a drug. It's not a mAb. I've made that mistake before, though. It's an inhibitor of the JAK kinases, which operate downstream. Interferon binds its receptor, and then there's signaling. So this will block the production of ISGs, basically. And so they show that if you have cells lacking the writer, remember what happens to HCMV when you lack the writer?
It does poorly.
It does poorly.
It doesn't get published.
And this drug, adding this drug will not make it poorly anymore. Will rescue it. HCMV replication.
Corrected its vocabulary.
Which says that interferon signaling is involved in the HCMV not doing poorly.
Right.
I'm not going to look at you anymore, Dickson.
Don't do that. No, don't.
And this does a little bit to help us understand where this regulation is happening. It's not that we're specifically regulating just the ISGs. There's something going on a little upstream of that with changes in interferon signaling.
Mm-hmm.
Right.
So this effect can be achieved using ultraviolet inactivated HCMV, where you don't get any viral transcription, right? You still get increased production of these ISGs in writer depleted cells.
Right.
So you don't need to have something about the virus coming in that's being sensed is triggering this whole thing. Could the ISG mRNAs be being regulated by methylation? Right. So one of the things that M6A modification does is cause depletion or destabilization of mRNAs.
Mm-hmm.
But the ISG, so they can do this genome-wide analysis of methylation and look at the ISGs, and they say they're not particularly enriched in methylation. So that doesn't seem to be what's happening here. And they're not decaying any faster when you're looking at cells with or without writers. So it's not the ISGs themselves. Well, maybe it's something upstream. And so they end up looking at interferon beta mRNA, and it turns out it's methylated at M6A.
And the reason to do that is because interferon beta is one of the gene products that kicks off the interferon response.
Right.
Exactly.
So it's going to stimulate the transcription of the ISGs. So now that's going to be the immediate upstream thing. Look at interferon beta and find that RNA is methylated normally in the infection. And that is, in fact, what's going on.
So they can see a specific methylation signal, M6A signal, near the stop codon of the interferon beta mRNA, and that's a typical place for this methylation to be. And if you take away the writer gene, it goes away or goes down, that methylation. So it's connected to the writer that we've been working with. They further validate the turnover of interferon mRNA. They look at the turnover of the protein as well. And so they're thinking that maybe this methylation is regulating stability of the message. And in fact, when they look at that, that's the case. When it's methylated, it's destabilized and degraded. And the cool thing is you can actually know which of the A's are methylated in the mRNA. And there were 3 of them in the interferon beta mRNA. They changed them to G, so they can't be methylated. And that gets around they're not methylated, they're not degraded anymore. They're more stable. Isn't that cool? I love that experiment.
Yes.
It's very cool.
Just changing a couple of bases. So now it's more stable. And when you infect these cells, you get a stronger antiviral response against HCMV in wild-type cells where the writers are present. And it turns out that interferon alpha is also regulated by methylation in a similar way. You may be thinking, what about other viruses? And they found some effects of these writers and readers on influenza virus, adenovirus, and VSV infected cells.
Do we want to wait and bring up the other paper results? Because the other paper looked at 2 other viruses and didn't see an effect.
Which ones were those?
They looked at HSV1, herpes simplex virus 1, and vaccinia virus.
It's interesting. So taking away the writer didn't have an effect on the replication of those viruses.
I'm looking to see exactly what they did. It was a supplemental data figure, but they referred to it in the discussion also. Depleting METTL3 or the ALK-BH5, that's the reader and the writer, right? Yeah.
Right.
Did not measurably impact replication of other DNA viruses, including HSV1 or vaccinia.
ALK-BH5 is the eraser.
It's the eraser. Yeah. Okay.
But either one.
And was it METTL3 or METTL14 that they were looking at there?
METTL3 is what they say here in the text of the other paper. Yeah.
So herpes and vaccinia. Oh, that's interesting.
I'm trying to remember if they gave a reason in the discussion. This might reflect differences in cell intrinsic antiviral responses. It does maybe, because HCMV doesn't have much host cell shutoff in contrast to the case for HSV1 or vaccinia, which gets strongly suppressed. The host synthesis gets strongly suppressed. So they did have a rationale.
Right.
So I'm trying to understand. So there is no effect. There's no depression of HSV or vaccinia yields when you reduce METTL, the writer, which means that there's no increase of methylation of interferons in cells infected with those viruses. So it's not that they're not being sensed, right? It's
right.
Well, you don't know. They could be not being sensed, or maybe those viruses have ways to evade this process.
Right.
Yeah.
Those are pretty good
mechanisms.
evasive viruses.
Yeah.
Yeah, vaccinia in particular has lots and lots of tricks up its sleeve.
So the implication would be that cytomegalovirus is not very good at evading sensing or downstream pathways, and that's why interfering with interfering with a writer depresses the infection because it's allowing stabilization of interferon beta.
Right.
So HCMV is being sensed effectively to turn on those interferon responses, right?
But something that happens in the HCMV infection process, which is not dependent on its, not dependent on transcription, is that it stimulates methylation of interferon alpha and beta RNAs, which destabilizes them and shuts off the interferon response.
So there is an interferon response when you infect with UV inactivated virus, right?
Oh, there is, right.
And it is affected by the methylation state for sure. So maybe the viral DNA is being sensed, right?
Right.
Yes.
As it comes into the cell, and that would not require infectious particles, right? It would just require DNA. Maybe the DNA is delivered to the nucleus, it's sensed, you get interferon synthesis, and then you get the same effect in UV inactivated virus, right?
Right.
So there are two things happening here. There's sensing of the genome, turning on the production of interferon and interferon-stimulated genes, and then there's induction of the methylase, the methylation system, the writers and the readers, which could be now. So, Brianne, is that known to be an interferon response, or is it a separate thing?
I don't specifically know of it as an interferon response, but the thing that I find really interesting about this is that we know that cytokines, especially interferon, are under really tight control.
Yep.
And I was talking to the high school students today about the fact that in immunology, we often think about Goldilocks. And how too much of something is bad and too little of something is bad, and you really often need to have the just right amount.
Right.
So, here we're seeing a host cell enzyme turning off a host cell immune pathway.
Right.
And the virus is actually stopping the shutdown. And you might say, well, why would the cell ever shut off interferon? Interferon's a good thing. We don't want to shut off interferon.
Oh, yes, we do.
Right. Right, and that's the point of too much interferon is also deleterious. We see immunopathology. So what's happening here is we're changing the turnover rate. We're kind of changing how stable that interferon is to allow it to be around a little bit more. We're saying, okay, now it's okay that we have too much interferon. We'll risk the pathology in order to get rid of this virus.
Right, and the virus is apparently stimulating this mechanism in a way that terminates the interferon response.
Exactly. But the virus isn't doing it by itself. The virus is changing host proteins in order to do that.
Right.
Wait, isn't the methylation response really a response to the interferon production by the cell to regulate interferon, not having anything to do with the virus?
Right. But it can be.
So the virus.
But the way we got here, the way we got here in the first place is they infected the cells, or at least the first experiment they discussed was infecting the cells with HCMV and seeing that there's all this M6A modification going on. So the virus, the infection has stimulated an increase in methylation.
Yes.
And so that benefits the virus because what it has stimulated specifically is methylation of interferon alpha and beta RNAs.
Right.
Which is going to that's the normal mechanism for for shutting down the interferon response.
Mm-hmm. And so it's basically destabilizing them. It's taking advantage of our normal breakdown mechanism.
Right.
But how do you know that's an effect of virus infection? You don't.
You don't.
And in fact, if you do with UV inactivated virus,
yes,
you still get the same. Well, I don't know if they induce it. They didn't actually look at that. You do get methylation, right?
And the other thing to notice is that in a lot of their figures, they're looking at 24-hour or 48-hour time points after infection.
Yeah.
And you can see interferon transcripts by like 6 or 8 hours.
Right.
Right.
So
the effects on methylation are much later than the induction of interferon.
Well, I don't think they show us the early time points to know.
No.
I think they've mentioned that multiple times, though. The differences in ISG only occur at later stages and are probably not related to differences in host recognition.
Okay, yeah.
So I'm just wondering if this increase in methylation enzymes that they start off the paper with, that they say translation, it's a supplemental figure. I don't have it right here. I wonder when that happens, if it's early or late. I'm just trying to figure out if it's just a cellular response. It's a like you said earlier, regulating the immune responses so it's not too much and not too little. And so that's a cellular response, and it just happens to have an effect on the virus.
But in this case, it's a cellular response that ends up benefiting the virus, so it's not a very advantageous response from the cell's perspective.
Well, I don't know about benefit because I just don't know if you can put it in those terms. Benefit depends on finding new cells and new hosts to infect and all of that stuff, and we don't know anything about that. In terms of titers, in terms of titers, that's all we can look at here. Yeah, there is a benefit if you take away the writers, right?
Yeah. You don't know what would happen with pathology in a whole organism.
Sorry, if you take away the writers, you inhibit. So writing is beneficial for the virus. Yeah, and that turning over. But, I mean, all immune responses eventually get turned off or modulated. Otherwise, they'd kill you, right?
But this one seems to be getting turned off. This one seems to be getting turned off a little bit prematurely from the host's perspective because it's allowing the virus to replicate to higher levels than it would if it didn't turn it off.
So that's why it would be interesting to know how the methylation system is coordinated by the cell with It must respond in some way to interferon production, right? I
mean, this sort of implies that it's responding to sort of the paracrine interferon.
Yeah. Yes, exactly.
But I don't know that it's what's the name of the drug that hits the JAK-STAT pathway?
Rituximab. No, go back. Ruxolitinib, yeah.
And that also sort of implies that this is part of the interferon response right there. But I don't have any outside additional knowledge about that.
So, I mean, it would have to be delayed somewhat because, it wouldn't make any sense to turn on interferon and signaling and then right away shut it off, right? Destabilize the interferon. And the reason I say that is, I don't know if you remember, we did a paper last year where it was shown that long non-coding RNAs regulate RIG-I sensing in some way, but that was a delayed response to allow RIG-I to sense and have interferons made and do their thing, and then it's turned off. So it would make sense to delay it until these proteins can actually work, do what they're supposed to do. But that's why I said at the beginning, the regulation of all this and its interplay with interferon system is going to be interesting to sort out.
Yeah, and in fact, I think this paper tells us more about, and this set of experiments tells us more about the immune system than it does about the virus.
Mm-hmm.
Yeah, one gets the sense they're just using it as a tool.
Right. I mean, they're using this as a tool, and they've found this new, apparently conserved mechanism for regulating interferon responses, which was, we didn't know that methylation of the RNA was a way to regulate the interferon. We knew like several other ways of regulating the interferon response. This is another one, which is now, I guess that's why it's in Nature Immunology.
That's what I was just going to say.
Yeah.
So now that's a whole direction of research on its own, is looking now at methylation in various immune conditions and autoimmune conditions and seeing what's going on.
So in fact, in the second paper, they look at introducing DNA into cells.
Yes.
They do infect with virus, but They also do experiments where they're transfecting double-stranded DNA into cells and looking at the interferon responses and the effect of methylation. So, yes, it's really the fundamental underlying principles. You don't even need a virus here, right?
Right.
Just put DNA in as they did in the second.
And the second one also has a kind of a nice model at the end where they show a balance, and actually a two-pan balance with the in this case, it's the eraser and the writer on each side of the balance, and then the amount of M6A and the interferon.
Yeah, right. So it's in fact, the legend says DNA or virus.
Mm-hmm.
Yeah. Also in this Nature Immunology paper, they show that this system operates in mice. Instead, everything's been done so far in cells and culture, and they make some knockout mice for the YTHDF3. That's the reader, right?
Mm-hmm.
The metals are the writer, Ys are the reader, and the As are the erasers. So mice lacking the reader, you get more interferon, as you would guess, because the methylation cannot be read to have an effect on stability of the interferon mRNA. So that's actually something that maybe people are confused about. The writers attach the methyl to the RNA, but it won't have an effect unless there's a reader to say, oh, there's a methyl on this RNA, a methylated A, and now we're going to turn on the RNA decay pathways or recruit decay proteins to this particular transcript.
So mouse experiment struck me as kind of a reviewer 3 type of thing.
Could have been.
It could have been, but it's nice in that it tells us that now we could start doing those experiments to look at immunopathology.
Yes.
That it is conserved here.
Mm-hmm. It's a lot of work, actually, to construct the mouse. And for a reviewer, you should make a mouse and do this. Oh, thanks. Another year of work, huh?
Right. They also made the mouse embryo fibroblast lines that were depleted.
Mm.
I mean, it's also possible that that's what they're doing to take the research to the next step.
That makes sense. It makes perfect sense. Yeah.
It's not necessarily that they were forced to do that experiment, but as I was reading it, I was like, oh, now we're in mice. I wonder why.
Mm-hmm. So they suggest that these observations could maybe unify some of the different phenotypes you see on the effect of depleting this methylation machinery on virus infection. You know, for some viruses, they replicate better. For some, they replicate worse. And they say maybe if you didn't think about the role of the immune response, it didn't make sense, but maybe now it will. So we'll see if people go back and do that.
They also, I like the second to last paragraph where they're talking about these relatively subtle changes in messenger RNA stability leading to strong phenotypes.
Yeah.
And that for the majority of genes, maybe a little bit of change in the half-life isn't a big deal. But for cytokines whose expression is really tightly regulated, then a little bit of change could make a profound effect.
Mm-hmm.
Right.
Yeah, absolutely.
Yes. So the genes and development paper has a lot of the same conclusions. As Kathy said, they looked at some other viruses and found differences. And they also look at DNA, not just delivering DNA virus to cell, but look at DNA as well. And
the thing that's sort of interesting there is that the DNA they use is vaccinia DNA.
Uh-huh.
And so they say that vaccinia virus does not have this. They don't see this effect in vaccinia virus, but they use really common vaccinia DNA for the stimulation.
So presumably it could have been any DNA, right? They could have bought the stuff
salmon sperm
DNA,
right.
But they have vaccinia around, so they made it. And
well, this is actually one you buy.
I could buy it, yeah.
I buy that same vaccinia 70 base pair DNA.
It's only 70 base pairs, huh?
Yep, 70 base pairs of double-stranded DNA.
And because there are no antagonists encoded there, then that's why you see a difference. Whereas when you infect, you see a difference. Okay. The second paper, by the way, is from Ian Moore's group at New York University School of Medicine. Rubio, DePledge, Bianco, Thompson, and Moore. And so they're both very interesting. And they were published the Genes in Development. When was that published? October
October 12th is when it was accepted.
last year.
Yeah, it was in the December 1st issue.
And the other one,
It was accepted November 7th.
and it seems like it's in the February 2019 issue. So methylation is hot.
Yeah.
Yeah.
Very interesting. What do you think, Dickson? Are you interested in methylation?
Of course.
Are
everyone?
Are parasites? Do parasites methylate their?
I had it for
mRNA?
breakfast twice while I was
bet.
in Europe.
What? Parasites?
No, methyls.
Are methyls sweet, sour, or?
It has a umami-like flavor. It's hard to describe, actually.
Look, what's the other word? Yes, go ahead. Go ahead.
Oh, I was just going to say, I did find a little bit more about the YTH domain family.
I told you Kathy would do it.
Well,
bet.
so it's a YTH is one of the potentially new RNA binding domains, and it's named after YTH 521 homology.
Right.
YTH. So what is YT 521? It's a novel RNA splicing related protein induced by hypoxia. And it just seems to have been given an arbitrary name, YT 521.
Yeah.
And so this is YT 521 homology like.
It's angiogenic.
It's not your title here.
No,
Does it have a lot of
not your
tyrosines
title.
and threonines?
probably not.
Or yttrium, thymidine, and hydrogen.
You know, it's because of you I look for these names, because you're always going to say, what does that mean? And if we don't know, we look silly.
Well, after you answered the question, I still don't know what it means.
Right. It's not like knowing the name tells you the function.
Sometimes you don't.
But you said it's associated with hypoxia, so I would associate with probably
It's a splicing factor.
angiogenic process.
Are you hypoxic at the moment?
No, actually, I feel pretty good.
Good. Yeah. All right, let's do some email. Kathy, can you take that first one?
Sure.
That was
Bill
a loaded
writes,
question.
This is not totally virology related, so I didn't think it was worth an official TWiV email. I sent it to my sister Kathy, but she thought otherwise. So, as a retired construction engineer, power plants and refineries, not bridges, here's my take. So, yes, this is Bill, my brother, the one who lives in Boulder. So, this is regarding the destruction of the Tappan Zee Bridge.
Yeah.
Right. I get several construction magazines and newsletters, so I was following along with the bridge construction and demolition. I estimate that 95+% of the old bridge was or is being demolished piecemeal. Perhaps the best general reference for this is this Wikipedia article, and he gives a link. But part of the eastern approach exhibited structural instability that actually delayed the opening of the second span of the replacement bridge last summer. Finally, it was determined that one span of the eastern approach was too structurally unstable to allow for piecemeal demolition to be done safely. So the explosive option was selected. I've attached one article. I think the thousands of tons of concrete and steel describes the total demolition of the entire bridge, not just the section that was dropped with explosives. That is only one truss section and 4 piers caissons. Here's a good video by the company that did the demo. They did plan to remove the steel from the river. And then he writes, I last went across the old bridge in July 2016 when I drove from Virginia to Maine. The new bridge was well under construction then.
Yeah, we've been watching it daily almost.
So the reason this is here, I had mentioned that I had seen a video of the span dropping into the river.
All right.
And I wondered why they did that and if they were going to clean it up and so forth. So it's interesting that it has structural, instabilities. I wonder if it did near the end of its life when cars were running across it.
But a pore formed at the end of it.
Yes, right. It was a VP2.
Right.
That's absolutely right.
Right. Anyway, the YouTube is...
Sorry, go ahead.
I was going to say the YouTube is pretty cool. I don't know if it's the same one that you saw, Vincent, but it's by the company that did the demo. And so there's all different angles of it, multiple shots of it.
I'm under the impression, by the way, that all of the structural components of the bridge, the old bridge, was shipped out to the ocean to make reefs.
Do you know that's all nonsense?
I heard that.
It's not nonsense, but I was listening to a podcast by a guy who studies, and he's studied since the beginning, fish that go around human-made structures, right?
Like shipwrecks and stuff.
And he was the first to do this. I think the first place it was done, they built those drill platforms off of Santa Barbara.
Yeah, the rigs.
Right.
And this guy was at the university, and he said, let's go out and take a look. And he found amazing fish.
Sure.
He got grants to study this.
Yeah.
And they actually now, so those rigs were built under a contract which said at the end of life, the companies have to pay for their removal. Now
They don't
want to remove them because
That's
they're going to disrupt the fish.
Exactly.
But a lot of people are like, no, make them pay for it. And it's an eyesore.
Yeah.
So it's a big problem. It does attract.
Oh, sure.
And you know, they throw subway cars into the ocean.
They sink battleships.
But they probably shouldn't be doing that, right?
Well,
This guy said they sunk thousands of tires off of Florida, and it was a disaster.
Tires are different.
They started leaching material.
Tires are different. Yeah.
Tires are different. The
metals are.
It depends. It depends on the material that's. I've gone diving on a number of artificial reefs, and it depends on the material and how it was prepared. And bear in mind, you know, we're sinking ships in the ocean all the time, just not deliberately.
Exactly.
Really?
This has been going on for a very long time. There
You
are a
should
whole
have
bunch
seen
of wrecks out there that just got there the natural way.
Do you remember the videos of all of the stuff they were dumping over the ships during the evacuation of Vietnam?
Sure.
Good Lord. A hospital's worth of operating equipment and stuff.
So should they remove the drilling rigs off Santa Barbara and kill all those fish?
No,
they would kill the fish. The fish would go elsewhere. Or they would kill the invertebrates that are all attached all over that stuff. No, I think those are fine to leave there as long as they've been decontaminated to some extent.
Eventually, all of that stuff will dissolve.
Well, the people in Santa Barbara want them gone because they look bad.
Well,
right?
I would remove the top parts, but I'd leave the other parts. Have you ever seen a map of the oil rigs in the Gulf of Mexico? You would be appalled. It looks like pepper was just sprayed on.
It's just that if you leave the lower parts, you have to put something there so a boat doesn't. Whatever. Right?
You cut them off deep enough. This is you cut them off deep enough that it's going to clear the draft of the ships. You chart them. You could put a submerged buoy for dive boats to anchor on.
Sure.
Because they're very popular scuba diving sites.
And there are the commercial fishing fleets that go out with customers that want to catch dolphin and stuff.
They're also very popular fish attractors, yes.
Yeah.
Yep. All right. We have a couple of links from Anthony. From Al Jazeera, distrust of polio vaccine plagues Afghanistan. So there's an article in Al Jazeera News. Many parents in rural areas suspect that polio vaccines are used for spying or contain questionable ingredients. So, and of course, Afghanistan, one of the countries where we have not been able to stop the circulation of wild polio viruses because we can't immunize everyone. So that's interesting. And then, bless you, Gesundheit. I don't actually want to bless you, but
there is an error. I think there's an error in the Al Jazeera article. They say after a fake polio vaccination program was used as cover to search for Osama bin Laden. I think it was actually a fake hepatitis vaccination.
It was Hep B, yeah.
Because they needed something that would provide cover for sticking people with needles.
Right. And then Anthony sends us an article on the origin of measles virus. It is in the Virology Journal, published in 2010. Origin of measles, divergence from rinderpest between the 11th and 12th centuries. And so we talked about this on Morbillivirus Had a Little Lamb.
Yes.
And I think it came up last week.
What was last week?
That was
that was filamentous things, right?
Mm-hmm.
Oh, so
it came up twice.
This was the original idea that measles was a result of a spillover of rinderpest from cows. But then in the paper, it was suggested that they both had a common ancestor, right?
So Rich talked about it one week, and then somebody wrote in and said it in a maybe more scientifically precise way, and Rich accommodated that when he read the letter.
Yeah. Well, anyway, this is an article which is open access, so you can look at their conclusion. Measles may have originated from virus of non-human species, caused emerging infectious diseases around the 11th to 12th centuries. Okay, so thank you for that.
They just came up with a different phylogenetic clock, and I haven't read the paper, so I don't know what the basis for this was, but that is the crux of the entire argument, I think, is how fast you think this thing evolved.
Well, in this paper, which is quite old, right? It's 9 years old. They say previously it was thought the divergence was thought to happen in the prehistoric age, but these new data suggest 11th to 12th centuries.
But what are the new data?
So I think there are even newer data which suggest it happened earlier than cows. And I don't know those data.
Okay.
Because Anthony writes, I'd been under the impression that the spillover occurred during the domestication of cattle. That's what I always thought. And that was said to us not correct, either in that paper or in a discussion. I don't remember. And Anthony also sends a link to Brianne's immunology course.
Nice.
Which we didn't put in last week, but we will put in. And finally, a Washington Post article, assertion is dismissed and no Holocaust matters. So Holocaust deniers have been sued. Why not anti-vaxxers, writes Anthony.
Why not is right.
Injurious denial of established fact. Yes. I would say if you're not vaccinating, you're injuring, right?
Yeah, climate deniers too, by the way.
Yeah,
especially if they're leading countries, populations larger than 300 million.
Dickson, can you take the next one?
Sure.
Ritesh
writes, Dr. Racaniello, excluding all the rest of us,
sorry,
of course.
I'm very sorry.
I enjoyed listening to the episode 531 of TWiV, where Dr. Patrick Moore talked about circular RNAs in KSHV and EBV. I think it's of the most interesting emerging topic in herpes virology or virology in general. I just wanted to point out that Dr. Moore mentioned RNA is encapsulated in herpes virions. I think a better word to use would be packaged instead of encapsulated, because it is still debatable whether RNA, in parens, host and or viral, is present inside capsids or in the tegument as argued by Dr. Roisman in this article. And then he gives a citation from an article from the NIH.
Yeah, I understand the point. The problem is that we often use packaging to mean in the capsid also. We talk about packaging signals that put the nucleic acid in the capsid. So I understand that it may not be in the capsid itself. I don't know what would be the best way to say it instead of packaging or encapsulating.
When you package something, isn't it going into a capsid?
I would say the circular RNAs are in the virus particle.
Yeah, included.
Included in the virus particle. Somewhere in there. Okay.
You might say it's a circular argument.
Alan, can you take the next one?
Sure. Paul writes, Dear Twivers, greetings from Brisbane, Australia. Muggy, very muggy here after some much-needed rain, virtually none at all in January. I have no memory of how we managed before air conditioning. Temperatures have been in the mid-30s, although southern states in Australia have been suffering with temperatures in the mid-40s and some outback areas breaking the 50s. And I may need to remind you that's 50 degrees centigrade.
Right.
Yes, I've been catching up on my TWiV podcast, so I'm a week behind. I was listening on my way into work today to the group's comprehensive discussion around African swine fever virus, illuminating as usual. However, I would like to pick up on a passing comment by Alan, I believe, who highlighted the difference in disease outcome between infection of wild versus domesticated pigs and the possibility perhaps one could engineer into domestic pigs the same resistance to severe disease. Vincent commented that he wasn't sure the genetics underlying the difference was known. Well, this has actually been done by the same institute that is famous for Dolly the sheep, the Roslin Institute. The work to generate genetically modified resistant domestic pigs was published in early 2016, so we've had the capacity to affect the impact of this disease for some time. While the science behind this is fascinating, it was centered on the identification and initial in vitro characterization in 2011 of polymorphic variation between wild and domesticated pigs in a gene called RELA, the VREL.
No, it's REL-A.
REL-A. Oh, I see. REL-A. Yes, REL-A. Okay. Yes, I get it. VREL reticuloendotheliosis viral oncogene homolog A. REL-A.
That's for Dickson.
I know it is. Yeah.
I have a plate of that, thanks.
The major component of the NF-kappa B transcription factor, a key component in the upregulation of immune regulatory genes. The polymorphic variation is a 3 amino acid difference, one of which appears key. Substitution of the domestic pig gene sequence for the wild pig gene sequence renders pigs resistant to African swine fever. Transgenic pigs were generated by older technology, but I imagine CRISPR would simplify the process. And then, of course, you would have CRISPR bacon.
Oof.
The real problem is not the science. That was my aside. Don't blame.
It was good.
Blame Paul for that.
I liked it.
The real problem is not the science. Is the fact that these transgenic pigs are, of course, genetically modified. Enter politics and hysteria, particularly in Europe, with their fundamental unscientific hatred of all that is GMO.
Right.
Yes, I have nailed my colors to the wall. The initial aim of researchers at the Roslin was to provide some assistance to pig farmers in Africa at the front line of the devastating effects of swine fever. At the front line of the devastating effects that swine fever can have on domestic pig populations in areas of regular viral spread. As you pointed out in the podcast, there aren't a lot of options other than quarantine and destruction of affected populations. But the GMO status of such animals has been a regulatory and political hurdle. I'm not up to date with where this law is now, but I can only hope that sense will ultimately prevail and solutions that are evidence-based are followed through to policy and implementation. I know, naive, huh? Still enjoying the evidence-based discussions and insights you bring to the virology community and the broader interested public. Keep up the great work. And then provides several links. One from National Hog Farmer.
I read that all the time.
I would, yes, it's on my coffee table.
Hey guys, don't knock this. I worked on Racaniello, and I used to read that publication.
You did.
Damn right.
Actually sounds like a really interesting magazine.
Pigs are us,
and I'm not kidding. Second to the paper, they've got the paper covering the modification, and this is based on earlier 2011 identification of the REL-A gene. And then gives a link to that. And
amazing that it's such a short. It's one gene that does.
It's one thing.
It's
yeah.
so cool. Now, I think I know someone who is, and you should know about this, who's making transgenic cows who will be resistant to trypanosomiasis.
Yes,
and that would be to give them to Africa so they can raise them there,
right?
And they will take them with open arms
and to destroy the entire sub-Saharan Sahel
because there'll be too many cows.
They'll overrun the place. That's yes. Right. They really don't want them. Actually.
But the pigs would help them as well, right?
Pigs would be a better deal.
So I think certain countries in
West Africa
would take these GMO or not.
In West Africa, the pigs
the problem
do
is that once they take them, their pork will be banned from European markets, which will provide a non-tariff barrier to protect the pig farmers in the EU who would prefer not to compete with cheaper pork from elsewhere. Yeah. So that's one of the big things with the anti-GMO movement is that it's really a lot of it is driven by farmers who want protectionist policies that would not otherwise be possible under modern trade regulations. Anyway, thank you very much, Paul. That's Paul Young, the head of school at School of Chemistry and Molecular Biosciences, University of Queensland in Brisbane.
Where's Brisbane?
Brisbane is in Queensland.
Yeah,
northeast.
I was going to say Australia.
Well, that's that part.
That's good,
yeah,
Kathy.
I kept my mouth shut.
It's a very good start. I've actually been to Brisbane.
It started by saying greetings from Brisbane, Australia. So
it's a lovely city. Yeah. It's a lovely city.
I just want to know what part of Australia.
It's in the northeast, eastern. Yeah.
Northeast.
So it is near Cairns.
Below Cairns. It's way below Cairns.
Okay.
Cairns is way up at the top.
Okay. Yeah. A few more links from Anthony. In a BBC poll, Alan Turing was chosen as the greatest scientist in a category that included Marie Curie, Albert Einstein, and Tu Youyou.
He was named. So this poll named him as the most "quote unquote" iconic figure of the 20th century. And the competition included not just scientists, but folks in other fields too, such as Martin Luther King Jr. and Nelson Mandela.
Turing was picked by the public during a live broadcast of BBC's The Greatest Person of the 20th Century. But I know that's Dickson. Anyway, Turing was important.
He was very, very important. I don't know if I would place him as being more important than some of these other folks, but that's kind of a silly question anyway.
But as Alan says, internet polls.
Yeah.
Really crazy. And Anthony also sends a link to another BBC article, which is a little interesting for us. So Spotify is spending many millions of dollars on a couple of podcast companies. The big one is they're buying Gimlet Media.
I'm holding my hand out, but no cash is landing in it. What's wrong?
Gimlet Media for like $200 million. I heard about this on a podcast this week. So meta, right? Gimlet Media was founded by the guy who used to do This American Life, or still does it. What's his name?
Oh, I know those
Ira
shows
Glass.
on
Ira
Glass. He's very good, actually.
Is it Ira Glass?
I think so.
Gimlet. Anyway, so he went off and raised money, and he has a bunch of podcasts. If you look up Gimlet Media, you will find them all. They have 70 or 80 employees. They have millions of dollars. They make money, though. They get advertising.
Yeah, sure.
So this is a profitable podcast company. In contrast, Micro TV is not a profitable podcast company. On the other hand, we don't have an NPR star person at the head there. And so this is something that will happen with these big podcast companies. And so Spotify just wants to get more media. They are headquartered right here in New York City, by the way. Alex Blumberg.
Yes.
That's the guy I was thinking of. Founded in 2014.
Michael Blumberg?
No.
Spelled differently.
No.
Oh.
So they have a lot of podcasts. And in fact, I was on one episode of one of them, and I can't remember, but they're all mostly highly produced. And
Sure.
so here, for me, I got an email from the lady who was the host of the podcast I was on. I can't remember what it was called. And she said, we'd like you to be on. And then a producer called me to speak for 15 minutes to make sure I could talk and be articulate, right?
That's right.
And then all these other people interfaced. And so that person, she didn't have to do any work.
Right.
And then she called me, they recorded it, and I'm sure all the post-production was done by other people. That's how it works.
Well, they had a machine.
Good to be the king.
And then it's all
good to be the
editing.
king.
It's all
That's right.
edited. They put music here and there,
No, it's professional.
Yeah, we're not. We have here a home-brewed podcast.
I hate to tell you this. We're the most professional. We all have professional
But
degrees.
we do provide interesting information.
Professional, we're trained in our fields, and we can communicate. And I'm very proud to be part of this group, I must tell you.
No, I am not proud. What's the opposite of proud? Embarrassed.
Right.
I'm not embarrassed at all. No.
Shamed.
I think it's great what we do, but nobody will buy us for.
Well, we
200
represent a very specific,
But you know
narrow
what?
subject.
We're not for sale.
We are not
That's right.
for sale. That's right.
We could be for rent.
Wait a minute. You really want to reconsider that for rent?
So
How
I
about
listened. I
long-term use?
heard about this on. Oh, so let me just go on for another minute.
Please.
I heard about this on This Week in Tech, which is the original podcast that inspired this show.
We were
right?
on that show together.
And This Week in Tech pulls in about $5 million a year in advertising, and he was lamenting that nobody wants to buy him. So,
You have to add a zero to him to get bought.
okay, let's do some picks. Brianne, what do you have for us?
So I have something that I came across while putting together one of my classes, and I think it's pretty fun. It is a card game called the Red Queen's Race. It's actually sort of a teaching tool where you use normal playing cards to put together a game where students can learn about host-parasite coevolution. So the students get to have 2 decks of cards that are the host and the parasite, and the suits are different genotypes, and you get to actually look at how evolution happens over a number of different generations. And I always struggle with talking to students about why we have things like such MHC diversity in hosts, and this really gets at why we have such diversity in genotypes. And so the students really like it and have a lot of fun with it.
Oh,
This is cool.
Very good. I like that.
Mm-hmm.
Dickson, you want to play sometime?
Sure.
Alan, what do you have for us?
But it always just goes to a draw, doesn't it?
Yeah, I think so.
And they just
I
keep
think
cycling back and forth.
That's right.
I think you're going to learn about the importance of genetic diversity.
It's like a game
Right.
of war.
Yeah, well, we can have draws. That's fine. Alan, what do you have for us?
I have a nifty little toy that I got for Christmas. So this has been sitting around for a while, and I've been enjoying it. It's just a little pair of inexpensive binoculars that are very portable, so nothing special about that. But what's really cool about these is that they focus very, very close. And maybe that doesn't sound like a big deal, but it's completely transformative in your experience of binoculars. So I'd read about these things. I thought, wow, that sounds really cool. So I asked for them for Christmas, and I got them. And I've picked the 6.5x ones. There's also an 8x, 8.5x model, which I think would be better if you're also doing bird watching. So you pick these things up, and you can use them like regular binoculars, look at things in the distance, or you can just look down at your feet and see the bugs crawling in the grass. And,
it's like a macro lens.
It's like walking around with a dissecting microscope in your hand.
Fabulous. Oh, good
and
stuff.
it's portable, and you can just throw it in your backpack. And I'm really looking forward to going hiking in the spring with this.
Wow.
Papilio is, I believe, Italian for butterfly. So would be
Japonicum. That's not. No, that's the Japanese beetle genus, I think.
Okay. Anyway, it's great for looking at bugs.
It shows people also bought the strap, so there is a way to attach a strap to them.
It comes with a strap.
Oh, okay.
So I don't know why people are buying a strap. Maybe they didn't get the memo that it comes with one, but it does.
Right.
Dickson, you have a pick?
I do, but I didn't get a chance to actually upload it. It's an artist at MIT, as I recall. It was on. I got the site from National Geographic. She actually works with scientists to help them envision their data. I have the link. Just let me get up from my seat.
Go ahead. Go get.
All right, I'll be right back.
I picked kind of relating to last week when we were talking about IPA and other alphabets, a chart. It's the history of the alphabet. And there's a blog post kind of article about it. And then if you go to useful charts, you can find more of these charts. And it turns out that the evolution of the alphabet chart is sold out, but they have a way you can download it for free. And then there's all kinds of other charts, like Canadian history, family trees, English royalty, Asian royalty at usefulcharts.com. And then the way to kind of shop through them, I think, is to go to the blog link at usefulcharts.com. And so that's how I found one that's a Venn diagram of the letters that are in common between Latin, Greek, and Cyrillic. So wow. It's just a cool set of charts and things.
Very cool.
Neat.
I didn't realize.
Yeah,
so
look very useful.
You look at some of the evolution. So A came from a character that looked like a cow or something.
Right.
Isn't that funny?
Mm-hmm.
And a fish became D and humans became E. Wow.
Yeah.
That's very neat. But I don't know why, right? Why would a human become
errors
E?
in transcription?
Or some sound that we've lost.
Right.
Who knows? That's cool. Very cool. My pick is my virology course lectures for this year. Every spring I teach an undergraduate master's virology course here at Columbia. I record all the lectures. I put them up on YouTube. So we are there are 4 up. There'll be 5 as of tomorrow. And number 5 is structure. And so related to today's paper. And so you can find them all on YouTube. You can get also all the slides I use and the reading and so forth at virology.ws/course.
You have 18,000 subscribers now on YouTube.
I do. I was trying to reach 25,000.
You're trying to reach 25. Remember? That's right.
Remember, we got a couple thousand, but could not get to 25. Now, I'm sure I didn't work hard enough. I didn't do things that I could have done, like
always blame yourself, right?
But now it's a pinned tweet, so it might happen.
Right.
I pinned my tweet. Kathy said
18,000 is pretty good.
Yeah, it's not bad. It should be 1 million. But
true.
People say don't get hung up on the numbers.
I have 152. Last time I posted a video was 2 years ago. That might have something to do with it.
All right, we have a listener pick from Peter. Brianne, you didn't read an email. Why don't you read it?
Okay. Greetings, TWiV team. I have a science pick for you, though it is probably better suited for TWIM. I have taken early retirement due to poor health and looking for a hobby. In addition to listening to Microbe TV podcasts.
Oh, how nice.
Decided that I would get back into microscopy. I found an informative source of information, which I thought I would share with you and your listeners. Microbehunter.com is an online microscopy magazine and blog, forum, and YouTube channel. It is targeted mainly at amateur microscopists, teachers, and students. It was founded by and is run by Oliver Kim, an Austrian science teacher. And he gives us a link to the YouTube channel, the blog and magazine, as well as the forum. Regards, Peter. So I'm excited to take a look at this for class.
Nice.
Cool.
I think that's great that in your retirement you're going to still listen to us.
Yeah.
Isn't that good?
And that you're into amateur
That's good.
microscopy.
You bet.
An amateur Leeuwenhoek. Right, Dickson?
Yeah.
Is that how you say it?
Mhm.
So what's your pick?
Leeuwenhoek.
Leeuwenhoek? It's Leuven?
I think.
Leuwenhoek?
Yeah.
Dixon, how would you say it?
Leeuwenhoek.
Leuven? It's Leuven. Kathy knows the phonetic.
As in
No, I don't.
As in Leuvenbrow.
But I think I found Dickson's link.
Good.
I Googled National Geographic, MIT, artist and scientists.
Bingo.
Perfect.
Bingo.
So the link is there, Dickson.
Okay.
I'm relieved.
Do you want me to describe it for you?
Would you please?
National Geographic reporter Catherine Zuckerman spotlights the work of research scientist Felice Frankel, a photographer who captures images that are intended to captivate and inform viewers about complex scientific advances.
Right.
Does that sound like it?
It does.
Frankel explains that the goal of her new book is to help scientists understand that beautiful images can engage the public.
Yes.
That's why we have Dickson on TWiV.
Really?
Beautiful image.
Yes.
Yes.
Thank God it's radio.
Yeah, it's a book. Felice Frankel on improving the visual side of science. I totally agree. That's why that's why Instagram works for science.
Pictures are
very
beautiful, though. The pictures are really gorgeous.
I follow someone on Instagram who she's a clinical microbiologist, and every now and then she makes a plate with funny things, and people go nuts over it. Right?
Of course. So,
So you click to the right on the link, and the very first thing is a really cool photo of Bacillus subtilis.
Look at that.
Right. So
People love it.
there's beauty in nature, and there's, that sort of thing.
And there's nature and beauty.
Thank you, Alan, for the corollary.
That's why I like to put pictures of science things on
Look, you were
Instagram,
just talking
right?
about structure.
Yeah, the structure is great. This week I posted 6 different virus structures on Instagram, virus receptor complexes, all picornaviruses that have been done, and you can get the coordinates and make very cute pictures. And so I posted all 6, and I said, does anyone know what viruses they are? And this is very funny. Matt Freeman, who is a virologist that we all know, said, Jimmy, Jeff, Sally, Annabelle, and Chad.
Lovely. How did he know that?
Wise guy.
What's the acronym?
If you knew Matt, you would know he's a wise guy.
Yeah, okay.
All right, that's TWiV 534. You can find it at Apple Podcasts, microbe.tv/TWiV. And if you listen to us on a player of some kind, please subscribe. We would really like if you subscribed. We understand if you don't want to give money or you don't want to.
No, we don't.
What other things? But please subscribe. If you don't want to answer our questionnaires, please subscribe. And that gives us numbers, hard, cold numbers. But if you would like to give us a bit of money, microbe.tv/contribute. And
Some
of course,
hard, cold money.
TWiV at microbe.tv is where you can send your questions and comments. Dickson Despommier, welcome back.
Thank you.
Thank you for participating.
I loved it.
Racaniello.org. And let's see, what are we going to spin today? Pick one.
Parasites Without Borders.
.com. Thank you, Dickson. Brianne Barker is over at Drew University on Twitter. She's BioProf Barker. Thank you, Brianne.
Thank you. It was great to be here.
Your microphone sounds good today.
Good. I'm glad.
Must be the internet's.
It sounds crisper than it did yesterday.
The interwebs. Kathy Spindler's at the University of Michigan, which is in Ann Arbor, Michigan. Thank you, Kathy.
It is.
Thanks. This is a lot of fun.
Periodically, I see cars driving around here with Michigan plates, and I always think of my Michigan podcasters, Kathy and Michelle Swanson. And I think, wow, if they only knew there was a Michiganite here.
A Michigander. It's a Michigander.
Michigander. Alan Dove is at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
If you want more of Alan's snark, you should follow him on Twitter. Because he's snarky there.
Yes, I am.
Did we say what happened to Rich Condit?
Well, he's not here.
I know. Why?
He's on vacation.
I don't know what happened to them. I never ask.
I see.
Off sailing somewhere or playing with grandkids.
He's going to sink a boat so that he can make a reef for fish.
Right. Scuba diving on it.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASV and ASM for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.